everyone. In our previous section, we have studied about the experiments that were required to conduct to understand the process of photosynthesis. What are the requirements for photosynthesis? What are the products of photosynthesis we saw through the experiments. Okay. So welcome back once again. Today we are going to discuss about the early experiments of scientists to prove the photosynthesis. Okay. The first experiment related to, okay, let me write early experiments on photosynthesis. Early experiments on photosynthesis. So let me tell you one thing. The first scientist who did research on plants and animals was, you know very well, his Aristotle is called as father of biology. What he said, Aristotle said initially that all plants get everything they need from soil only. Okay, that was knowledge at that time. There was no like microscopes were not there. There was no idea of elements. So depending upon the knowledge of their time, it was their obvious reasoning that plants get everything from soil. But later on, scientists became aware, and the scientist who did first research on plants after Aristotle is von Helmont. What did von Helmont say? So this is a scientist, von Helmont. What is his experiment? Let us see his experiment. He took a pot, some soil in this pot. He measured the weight of the soil. He took a small trunk of a small willow plant into this pot. He even measured the weight of this trunk. The soil weight is, you can see, 200 lbs. Approximately it is equal to 90.76 or 77 grams. Okay, approximately. I am not saying exactly, something around 72 to 76. Okay, 90.7 grams. He took the willow plant of 5 lbs, 2 point some grams. So the total weight taken was 2.5 lbs at that time. He left this setup like this. See, what he covered over this plant is a small metal structure, so that only water goes inside. Rainwater can go inside or any other water can go inside. And he left it for 5 years. Okay, for 5 years he allowed the plant to grow like that. After 5 years, the difference that he noticed was a 5 lbs plant, that means approximately 2 grams, plant grew into a structure approximately 2 grams. Okay, this is 90.72 approximately. So total is 92.72. But after 5 years, the difference he observed was the plant grew to 70 plus grams. Okay, what the plant grew to was 70 plus grams. After 5 years, the plant's weight was 70 point some grams, and the soil weight is as it is. There is no difference except 90.72. It decreased to 90.66. A little 0.6 grams difference in weight he saw. Okay. So that is a very minute difference in soil's weight. If you see the plant's weight, there is a lot of difference. If you come to the conclusion that Aristotle's concept is correct, then soil should be used up, approximately 70 grams of soil should be used up. But that's not happening here. What is happening here? The plant's weight increased by approximately 68 grams. The soil weight came down by only 0.06 grams. So what conclusion did Von Helmont made is, plant does not grow only because of the soil that it gets. Plant does not simply grow because of soil it gets, but plant grows because of water it gets. Plant grows because of water it gets. Water it gets. Okay. So this is the conclusion of Von Helmont. Next experiment related to photosynthesis was conducted by another scientist, Joseph Priestley. Joseph Priestley. Okay, you might have studied about this scientist even in your chemistry topics. Okay, what does Joseph Priestley do? He took one candle, he placed a bell jar. Bell jar is a closed container, there is no chance for any flow of air from outside. So what happens? And you know this what happens. If you keep a candle in a closed container for some time, oxygen is required for burning. So after some time, the oxygen that is present in the bell jar was used up and the candle put off. Okay, but he did not have any idea about oxygen at that time. When Priestley was conducting experiment, oxygen was not discovered. So he did not understand the reason. After some time, he took willow plant and candle, he kept it under the same bell jar, but the candle did not put off. So he got some rough idea of what is happening. Next, what did he take? He took one rat and candle under the bell jar and closed that bell jar. After some time, he found that rat is found dead and candle was put off. Now what he did? He took the mint plant, he took the candle and rat, this time living rat and burning candle under the bell jar. And what he found is, he found both of them survived, rat survived, candle survived. So these are the experiments that are conducted by Joseph Priestley. So what was the conclusion that he made? Joseph Priestley concluded that animals and candle is making are making air impure, are making air impure, while plants are making it pure. Plants are making air pure. So that was the conclusion made by Joseph Priestley. He did not say this is oxygen, this is carbon dioxide, nothing he said. Later, the name oxygen was given by another scientist, Lavoisier. Lavoisier is a scientist who named the gas that is evolved from plants, which is utilized by animals or candle as oxygen. Okay, so this is the discovery of oxygen. Now, here it was proved that water is required for plants. Here it is proved that oxygen is released by plants. Next experiment was conducted by Jan Ingenhouse. So the next scientist who conducted experiment on plants is Jan Ingenhouse. When he placed an aquatic plant in sunlight, the green parts of plants are releasing bubbles only when they are exposed to sunlight only. When there is no sunlight, when they are kept in darkness, they will not show the bubbles. So what he concluded is sunlight is required for photosynthesis, required for only green parts of plant, green parts of plant to perform photosynthesis, This was given by the scientist Jan Ingenhousz. Okay. Later, another scientist, their names are Pelletier and Cavento. Pelletier and Cavento are two scientists who named the green substance that is present in plant as chlorophyll. Chloro means green, Fill means leaf. Okay. Since there is a substance in leaf which gives green color to the leaf, that green color giving substance is named as chlorophyll by scientists Pelletier and Cavento. Okay. But where is it present was given by another scientist. His name is Julius von Sachs. Julius von Sachs is a scientist who observed the green leaves and found that all the green color substance are located in particular region called chloroplast. Okay, so we know that chloroplast is an organelle. In this organelle we have the structures called chlorophyll, but that idea was given by Julius von Sachs. Okay, later on another scientist, his name is Daniel I. Arnon. Daniel I. Arnon is a scientist who could carefully isolate the chloroplast from the leaves without damaging. So his name is Daniel I. Arnon, and what he did is isolated chloroplast. Okay, one more thing was discovered by this scientist. His name is Engelmann. What Engelmann discovered is Engelmann is a scientist who discovered which region of sunlight is very suitable for photosynthesis. What he did is, he took a Spirogyra plant. You know what is Spirogyra? It is a aquatic organism, microscopic organism which is found in water bodies. This plant he exposed the sunlight, or he exposed it here, brand be shown, but he exposed it to sunlight. He allowed the sunlight through this prism. Okay, when light passes through this prism, what happens? You know very well. Light is split into 7 different colors. Okay, he allowed this light, which is split into 7 different colors, to fall on this Spirogyra plant. Okay, Spirogyra is a microscopic aquatic plant, and in this setup he allowed some oxygen sensitive bacteria. Okay, oxygen sensitive bacteria means which is very sensitive to oxygen. Okay, oxygen sensitive bacteria. Oxygen sensitive bacteria which goes to the area where there is more oxygen release. So these are the white color dots you can see. These are oxygen sensitive bacteria. The oxygen sensitive bacteria accumulated in 2 regions. Can you guess which region of visible light are occupied by this oxygen sensitive bacteria? Is it violet, blue, indigo, blue, or green, yellow, orange, red? Most of them accumulated in 2 regions. What are those 2 regions? They are accumulated more in blue region and red region. Okay, so this experiment proved that blue and red regions, there more oxygen is released. Okay, that's why they accumulated there. That means there more amount of photosynthesis has taken place. This Spirogyra performed more photosynthesis when it received blue light, when it received red light. Okay, that is why more oxygen sensitive bacteria accumulated in that region. Is it clear? So this experiment proved that this photosynthesis occurs mostly in two regions, that is blue and red regions. Blue and red regions, more amount of photosynthesis occurs. Okay. So Engelmann is a scientist who proved that blue and red regions of light, red regions of sunlight. So in this region only more photosynthesis occurs. More photosynthesis occurs. Lastly, one more scientist. His name is C.B. Van Neel. What did he say? C.B. Van Neel is a scientist. C.B. Van Neel is a scientist who gave the first formula for photosynthesis. What formula did he give? CO2 plus H2O gives rise to CH2O plus water plus oxygen. Okay, this is a formula that is given by C.B. Van Neel. He did his experiment on various organisms like Claudophora, on purple sulfur bacteria. Which bacteria? Purple sulfur bacteria. Okay, when you did experiment with purple sulfur bacteria, this is a chemosynthetic bacteria. That means it uses chemicals for making its own food. It is a type of autotrophic organism which does not require sunlight. It requires chemicals to make its own food. What is the chemical that it uses for making its own food is H2S, hydrogen sulfide. Okay, hydrogen sulfide is required for this purple sulfur bacteria to make its own food. This is a special type of autotrophs where the organisms use chemicals for making their own food rather than sunlight. So this organism, when it used H2S, the final compound that released out was not oxygen, but instead of that elemental sulfur came out. That means here whatever compound is it, that is coming out. So this carbon dioxide is not releasing its oxygen outside, but it is utilized to make some carbohydrate molecule. So based on these all reasoning, he gave this formula. Okay. So according to him, what he said is that is true. Nothing is wrong. What he said. What he said is hydrogen is whatever molecule that hydrogen is holding, that molecule will come out as a free gas. Okay, and carbon dioxide that is taken in is used for making carbohydrate molecule. Water molecule is produced during this entire process. So based on that, he gave this first formula for photosynthesis was given by C.B. Van Neel. So these are the different scientists. We have discussed about various scientists who did experiments on photosynthesis. Like we started with von Helmont, Joseph Priestley did experiments, Ingenhousz did experiments. Later on we saw the chloroplast discovery was done by Pelletier and Caventou. Then Julius von Sachs said that chlorophyll is present in chloroplast. Daniel I. Arnon extracted the chloroplast from the leaves, and we also saw the experiments that are conducted by C.B. Van Neel. What C.B. Van Neel said, the formula that is given for photosynthesis, and the experiments of Engelmann that prove that blue and red regions of the blue and red regions of light are suitable for photosynthesis. So these are the different experiments that are conducted by various scientists to prove photosynthesis. In our next class, we'll discuss about chloroplast and pigments that are involved in photosynthesis, and also we'll discuss about the various things like photosynthesis, light reactions in photosynthesis, dark reactions in photosynthesis. So tomorrow our discussion is exclusively on photosynthesis. Okay.
Okay children, that is it for today. I hope you have followed. Keep yourself safe, healthy and also study well children. Bye.